My name is Matthew Kaiser. I am a technical sales scientist at DNASTAR. I am the person here that will help customers with their next-gen workflows. I provide webinars like this for a group or individually. And today we'd like to cover a specific workflow. This will be bacterial genome assembly and manual gap closure. And so we will have a few PowerPoint slides, and then we'll go into the software and look at a couple of assemblies, and then do the downstream steps of closing gaps off and finishing genomes. So, of course, we're located here in Madison, Wisconsin. The weather looks nothing like this today. We do have a snowstorm that's ongoing here, so hopefully we'll be able to get through the next 45 minutes to an hour without any interruptions. But if you do hear sound quality issues, which could occur if our bandwidth narrows at all, please let us know, and we'll try to fix that. So just a little bit of background regarding DNASTAR. We are a pioneer in genomics. Our founder is Dr. Fred Blattner, who sequenced the E. coli genome back in the 1990s. This is just a little snapshot of the science paper from 1997, where he sequenced E. coli K12. And some of the folks that are on this paper still contribute to DNASTAR, so we really have a nice resource in our scientists here that have many years of experience developing sequence assembly software, and then also developing the downstream tools that you need to visualize assembled data, do annotations, work through gap closure. So our software really excels in these areas. Our software really is always kind of focused on desktop solutions. We do have some offerings on the cloud now, and that will continue to expand in the future, as well as our assembler can run on server software or on servers as well. So DNASTAR software is research grade. It's suitable for publication. Going all the way back to the mid-1980s, DNASTAR has been the top cited bioinformatics software. And so, while the software is easy to use, it's still very powerful and will produce results that can benefit you in your research. And so just kind of a summary, our engine assembly software, it's 64-bit. It runs on Mac, PC, and Linux. It is agnostic to platform. We can assemble Illumina, Ion Torrent, 454, PacBio data. It can be single-end data or paired-end or mate-pair data. And then the analysis tools that we'll look at today are in SeqMan. We also have analysis tools in our ArrayStar. These three softwares, SeqMan Engine, SeqMan, and ArrayStar, are bundled together in what we call our Genomics Suite. And so for those of you that already own the software or are considering the software, we do have these separate programs that are bundled together. The capacity is very high for templated assemblies. We work with human genomes and exomes and multiple genomes and exomes together. Our Denovo assembler that we'll look at today is generally used for microbial genomes, eukaryotic transcriptomes, and smaller eukaryotic genomes. So we're not really talking about large eukaryotic de novo assemblies and closing those gaps. Those data sets are still in the realm of Linux clusters and non-GUI interfaces. The cost for the software is low for academics. Genomics Suite is under $5,000 for both the assembly and the analysis. And of course, the support that we provide, we strive to have the best possible support for all of our customers. And so that means frequent webinars like this once a month to cover the more popular topics. We also have over 100 videos on our website that are roughly 5 minutes or less in length for specific topics. And then I personally do webinars with customers one-on-one. Oftentimes customers will send me a data set, and we'll look at the data set together, do a webinar, and go through the workflow so that I can show you how to use the tools and the software for your particular project. So one of the questions that we have it often regards the power of the software, and really, is it going to be powerful enough, on your desktop computer to handle the type of projects that you want to do? For reference-guided assemblies, we can do whole human genomes in under 24 hours, and that time will actually shorten quite a bit in the next release later this year. and that will dip under 20 hours for sure for that benchmark time. And exomes, which again are very large sets, a couple of hours per exome, and then the smaller genomes, fungal genomes and smaller plant genomes may only be a few minutes to do the reference-guided assemblies. De novo assemblies are a little bit more difficult to assess. Assembly time isn't always the best metric. It's part of the equation, but really you want contig accuracy. You want big contigs that are as accurate as possible. And so we look at a number of different things like the contig N50 for contig size, assembly time, but it's really after you start putting the genome together that you can figure out which assembler is best and which one isn't so good. And those are some of the steps that we're going to look at today, are the gap closure steps, where you really need accurate contigs before you start doing gap closure. If your contigs aren't good and you start doing gap closure, you're just putting together bad contigs and actually creating more work down the road. And so SeqMan Engine is a very stringent de novo assembler. It's great for genomes. It's great for transcriptomes as well. So the different workflows that our software supports, some of the main workflows are genome resequencing, exomes. With both of these workflows, we can multiplex and have multiple samples in a project. We have some webinars from 2012 that cover these topics, as well as RNA-seq, again, another hour-long webinar on our website that covers that topic. De novo transcriptome, and then what we're going to look at today is really a continuation of two webinars that I did in 2012, and that's the automated bacterial genome closure and de novo genome assembly. And so we're going to today we're going to take both of these workflows all the way to completion, and so we'll look at kind of both, and we'll talk about the tools that we have to manually close gaps and how we would do that. So one of the first questions that we have, so if you have a genomic data set, is whether or not to choose a de novo or a reference-guided approach. And there really isn't a right or wrong answer all the time. A lot of times there's an overlap here of which approach might be best. With a de novo approach, we're going to use SeqMan Engine to assemble reads into contigs, to cluster them together. And the contigs then, using mate pair or paired end information, can be ordered into scaffolds. And with really nice data sets, so if you've got, the ideal data set would be, 1,000 base pair plus long reads with a very low error rate. And if you have that, you can get de novo assemblies, all the way down to 25 contigs to reach the genome length. Of course, most people don't have that type of data. PacBio isn't quite accurate enough. It's long enough, but we don't quite get the accuracies. Nanopore hasn't hit the market yet. But we have long ion torrent and long Illumina data that's, a couple hundred to 300 or even 400 base pairs long. And if you've got, or 454 data, so if you have next-gen data and you have good read length, you can get an assembly where if you take the top 25 to 100 contigs and order them in a scaffold, you get your entire genome length. So you've got somewhere between 25 and 100 gaps to close. Now, if you do an assembly of your data and you've got 1,000 contigs, usually you want to continue to improve that assembly. You don't want to start gap closure if you've got 1,000 gaps to close. It just simply would take too long. You'll save time and effort by either adding new sequence data or adding better data before you start doing that gap closure. And I cover this workflow of setting up these de novo assemblies in the webinar Denovo Genome Assembly and Lasergene Genomics Suite from August 22nd, 2012. So that covers, in detail setting up a de novo project. Now, if you have a reference sequence that is close to your sequence strain, you're definitely going to want to try out a reference sequence, or you might have, a handful of reference sequences. You're not sure which one is best. It's worth, running an assembly that takes a couple of minutes to find out if one of these reference strains is close to your sequence strain. Our software also has the capacity to align sequence data to a database of reference genomes. So if you actually don't know what the best strain might be, you can align it to several hundred genomes at once and find the best or closest match in a bacterial database. Once you find what you think is the closest strain, then you can use this new workflow again, this is a webinar from December 5th, 2012, and it uses some new algorithms that we've added to SeqMan Engine that will align sequence data to a reference strain, identify structural variants, so places where there's insertions, deletions, inversions between the sequence strain and the reference strain, and then auto-resolve as many of those areas as possible. And so, these close reference genomes, there's not an absolute number, but what we mean by close is that when you do the alignment and then you look at the number of structural variants, hopefully it's less than 100. It might actually be, maybe less than 200 or 300 structural variants, and that means that they're going to be close enough. At some point, though, it's better to do a Denovo assembly. If your strain is too divergent from your sequence strain, then a Denovo assembly may get you closer to a completed genome than using the reference sequence. So that's where the overlap is, and in many cases there's no clear answer. You kind of do both. You do some Denovo, you see if you run into anything difficult, you try the best reference sequence, and then you make your decision down the line after looking at both assemblies. So so we're going to look at both approaches here. Once we get to contigs and scaffolds, then the manual, and you've taken all the auto tools as far as you can and the auto gap closing algorithms, then you're really doing manual gap closure, and the steps are the same for either workflow. So there are some requirements here. So for doing gap closure and using our software to do gap closure, it's really essential that we have either paired end or mate pair sequence data. And the reason for this is that most genomes have a number of repetitive elements. E. coli has anywhere from probably 10 to over 100 IS elements that are repetitive throughout the genome. And it's at those locations that de novo contig assembly generally ceases. You get to a point where reads map multiple places, the assembler doesn't want to make an error, and so it ends the contig at that repetitive element. And so that's why when you do a de novo assembly, you don't just get one big contig generally. You get multiple contigs. It's not always repetitive elements, but most of the time that's what's causing them. And so to put the contigs in the correct order, you need to have pair information that can span those repetitive elements. So if it's an IS element that is 1,000 base pairs or a little bit more than 1,000 base pairs, ideally I would have mate pair data that where the forward and reverse reads are more than 1,000 base pairs apart. That allows some uniquely placed pairs to span the repetitive element, and then to place two contigs next to each other around those elements. Likewise, with the reference sequence, the automated gap closure workflow, we use the pair information for scaffolds, but we also use it for resolving insertion sequences. And again, this is covered in detail in the webinar from earlier this month, or earlier in 2012. And so the AGC workflow will use that pair information to fill in these novel insertion regions. And so for either workflow, you really have to have this pair information. The automated genome closure workflow requires a reference genome, and I say about 300, ideally around 100, and we can stretch that to maybe up to 300. But the strain has to be close enough that we're not breaking the reference sequence up into too many tiny little pieces. Again, if we're breaking up the reference genome into many little pieces, a de novo assembly may give us better results, especially if it's a less repetitive genome. Then we might get bigger de novo contigs. So there's quite a bit of variability just on the nature of the genome as to which approach works best. Both approaches then, once we have contigs and scaffold, require the researcher to figure out what the gap-filling sequences are. And so when we have a scaffold and we have the gaps, we're going to use different things like PCR, different approaches. We might blast the edges of contigs. We might look at the unlocated contigs that we couldn't place in scaffolds. And we're going to have to do analysis on those regions and figure out, what each gap is caused by. And of course, we're going to suspect repetitive element. So if we can find a potential repetitive element that fills the gap, those are the tools that we're going to look at today, and how to use those tools properly. I can't tell you how to actually identify what the best gap-filling sequence is. That really requires just your knowledge of the genome, and trying some different trial and error approaches to figure out what that gap-filling piece is. And then really the other requirement is having a program like SeqMan Pro where we can look at everything, visualize the assembled reads, the contigs, to help organize the project as we do the gap closure steps. So at this point, we'll jump out the PowerPoint and take a look at the software. And so we'll start with SeqMan Engine. And again, SeqMan Engine is the starting point where we're going to set up these assemblies. I'll go through this in more detail in the webinars that I did previously. We're going to do a genome assembly here, Denovo, name the project, save the output as a SeqMan output. And the SeqMan output is fully editable. And so we need to have this editable project so that we can split contigs, merge contigs, add new sequence data to fill gaps. So we have to have this editable format. The BAM format is not suitable for gap closure. It's suitable for looking at the projects, but it is not suitable for gap closure. I also don't really recommend ACE format anymore. ACE format, I know some 454 customers still use ACE format. It's just not efficient enough for next-gen projects and just the structure of the file. So you really want to stick with the SeqMan format. And we can load in some sequences. Again, this is going to be Illumina and paired end data. This isn't ideal paired end data. This is 500 base pair inserts that I have here. But it still will work. And we're just going to stick with quality end trimming and default options. And when we get to this assembly options, I entered in my genome length. And there are some changes that I might recommend here. We want to have, when we're doing gap closure steps, we want to have the most accurate contigs possible. And it's okay, I would prefer to have smaller, more accurate contigs than larger contigs that have a false join in them. And so the minimum match percentage is a single parameter that we can adjust to affect the stringency of the assembly. With Illumina data, we already assemble at a pretty high stringency. 93% is pretty high. But I might try 95. I might go a little bit higher and see, do I have my contigs a lot smaller or just a little bit smaller? If they're only a little bit smaller, I may go with 95 to make a more stringent assembly. And of course, the different platforms, Ion Torrent, the default is going to be 90%. 454, I believe, is 90 as well. And so this is the one parameter I might tweak a little bit when I'm doing a Denovo assembly. Another thing that I change are the small contigs. And I increase the minimum. By default, the minimum is 0 for a minimum length, but and this is essentially culling out the smallest contigs from the assembly. And depending on your sequence data, you might get a lot of little tiny contigs forming. And a lot of times it can be noise from a contaminant that's in the sample, there's just different reasons why you can have some small contigs. But I'm going to remove contigs that are less than 250 base pairs. And what that does is it dissolves those smaller contigs, and it makes the project more manageable. I'm not going to use contigs this small for gap closure. I'd rather just dissolve those back into the unassembled reads, into the bin, and maybe look at those later. And so for some projects, this is very important. Denovo transcriptome, very, very important to do this. You don't want 20,000 contigs that are 100 base pairs long, so you want to call those small ones out. Then we're ready to assemble, and of course we can open the project in SeqMan Pro. And so in SeqMan Pro, then we get a couple of windows are open by default. We get the project report window, and then we get the list of contigs window. And the list of contig window, when you first open this project, will just be a list. It'll say unlocated contigs, and you'll have just a list of all your contigs. So it'll look just with this header, and there won't be any scaffolds. So it's one of the key differences with our software versus some other softwares is that we do not auto scaffold from the start. We start with contigs. It allows you to look at contigs, possibly edit them, split them, break them if you think there's false joins. And then we have a separate step that creates the scaffolds. And this is a project that, this is some older ion torrent data. And so there's quite a few scaffolds. It's a good data set to show for this kind of a demo. And so to create the scaffolds, what I do, once I'm convinced my contigs are all good enough, then I can go to Project and order contigs into scaffold. And so this is an algorithm then that reads the pair information and builds the scaffolds based off the pair information. So once I do that and I have all my scaffolds, then we have another algorithm, and the algorithm is to look within the scaffolds and align contigs within the scaffold and look for end overlap between adjoining contigs. And this is called align contigs in scaffold end to end. So it's a very specific type of algorithm. And the reason for this is that, again, with E. coli, many of these contig ends may end with some of the IS elements, just, a few base pairs of IS element on the end of the contig. And if I just throw all those contigs in one pot and just say align them, this algorithm right above, what can very easily happen is we could have contigs from all different regions of the genome joining together, misjoining. And so we definitely don't want to use align contigs in this case. We want to use a constrained aligner and look for overlap only in the adjoining contigs. We don't want to flip contigs over and reverse complement. We want to keep them within the boundaries that the pair information has specified. So we click this, and I'm not doing it live time here, so it takes about 5 minutes or so to do all the end aligning. But if we look in the report, you see where it says all the merging. And this is the list of the end aligning from, and you can see I'm just scrolling through. We've merged, this is gap closure then. So we've got our scaffold. So this is an auto algorithm, and you can see there's probably 100 or so. Now there's a lot of contigs in this assembly, so we get lots of merging here. And so this is an algorithm that does some auto gap closure within SeqMan. Once we do this, we have scaffolds, and you can see the scaffolds contain multiple contigs, and we can highlight the scaffold or double click on it, and it will bring us to a strategy view. So this is a scaffold strategy view, and I'm just going to zoom out a little bit here, and we can see here's contig 281 and contig 204. The scaffold, I should point out, we have a length of the contig within the scaffold, the number of sequences, and then the position. And so scaffolds assign position values to each one of the contigs. And so this position value is what determines which contig is next to which one. And so now we have, in this case, two contigs, contig 281 and contig 204. And if I zoom in here a little bit, we can see there's a little gap between them. It doesn't show up at this scale terribly well, but and I can zoom out. So there's a little gap between the contigs. And then in the bottom of the view, I'm only showing the pair data, and this is pair data that is 10 kb and about 4 kb, so it's two mixed types. And I'm only showing the blue pairs. So there's a key over here that allows us to make different types of reads visible in the strategy view. So again, it's a part of the really nice interface in SeqMan Pro that allows me to just look at what I need to look at to make the decisions on scaffolds. And so in this case, I'm selecting the consistent and group. It's the blue pairs. And so this is showing me mate pairs that where a forward reverse is on one contig and its pair is on the other. And so these are the pairs that SeqMan uses to build the scaffolds. And so you can see for these two adjoining contigs, there's a massive amount of pair information here. So it's very, very likely that these, in fact, go together. And we can look at, I can zoom in a little bit more. So I can toggle between show the sequence names or not to show them. Oops, I zoomed there. And so now I'm zoomed way in, and I can scroll, and there I can see the pairs. And if I zoom in further, you'll actually see those are arrows. I'll just kind of move over to that spot. So we have little arrows here, and there's a few other things we can do. We can get some sequence information about those arrows, and so I can actually see my forward and reverse, and a suffix is added, whether it's a forward or reverse pair. And so it's a nice interface for seeing, I've got this scaffold. How much pair information do I actually have to build the scaffold? If there's a problem with the assembly, or if a scaffold is put together sometimes with a small amount of pair information, I might decide to remove a config from a scaffold, or to rearrange and make edits within the scaffold. So again, it's a great view for just confirming that my contigs are good, my scaffolds are good, I've got lots of pair information to support that. Once we get to this point, however, with a Denovo assembly, we have to, essentially push up our sleeves and start to do some of the more difficult work. And that's, if I want to close this genome, I need to close all the gaps that I see between the contigs, between all the scaffolds. Now, of course, a project like this, I've got a lot of scaffolds. I'm going to go back and get some more data and try to reduce the number of contigs here as much as I can afford to buy more sequence data. And once I get that number down to what I think is workable, and it's definitely going to be below, my target would be 100 contigs or less, then I'm going to start the hard work, and that's closing these gaps off. Now, to figure out what's in the gaps, that really is the key, and there's, and it's different for every gap. Different researchers will use different techniques. One possible technique is to, and it's something that you have to do if you've got a truly novel, if I've sequenced something that is completely novel here, and I blast and I don't get any good blast hits, I may have to do directed PCR to actually PCR across gaps and create amplicon fragments and then bring those into SeqMan to close the gaps. If I want to do that approach, I will take my scaffold sequence and send it over to our Seq Builder. And we'll give this just a little bit. So I'll take this whole scaffold that's going to be fairly large and send it over to Seq Builder. And then I'm going to make the views here. Now this isn't intended to be a tutorial on Seq Builder use, but I'm going to make my primer list visible here. And so I have a linear map in Seq Builder. And of course we have if you haven't used Seq Builder, we have multiple demo videos on our website to show you, how to use Seq Builder. There's a lot here that we're just going to look at a small part of it. But I have a linear map here, but none of the features are displayed. And to use Seq Builder, I can say, let's look at certain feature types. And excuse me, one of the nice things with the connectivity between the different Lasergene modules is that SeqMan actually creates feature types related to the assembly. And so I can look at things like show me where contigs line up, or show me, it's created a feature where I have different types of coverages. If I'm doing gap closure, I'm really interested in, show me where the gap regions are. And so these, again, are created by SeqMan and displayed in the Seq Builder, and they're meant to kind of facilitate this sort of work. And so I can show gap sequences, and I can zoom in on them. So there's a gap then in so this is a scaffold sequence. And if I went and looked at the sequence view, I think there's one N or so. Let's see if I can get there. 110. I guess I can't easily show that here, but there's the scaffold sequence is going to have a single N place there that shows that there's actually a gap here. So if I want to design PCR primers, what I'll do is I'll just say, highlight my gap, go to the priming menu, and then create primer pairs. And this is going to bring up, let's see here. we call this the primer workbench. And so it's going to design primers then that span that gap. So I'm going to say, choose primers that are optimally within 100 base pairs of the region that I selected. And I can set conditions here. I won't go through all these details, but I can, typical primer designing kind of tools. And so now I get this workbench that will show me, the primers that I designed, and I can go in, here's a primer list, so here's my best set of primers that we found here, and I can go back to a linear map, and there's my targeted gap sequence. I can right-click here and detach, so it tells me which contig the gap is between. So it's really nice for saying, okay, I have some primers here that, if my gap is small enough and my PCR amplicon is large enough, I should be able to generate a fragment that can bridge the gap and connect contig 540 with contig 537. The primer design interface has what I think the best feature is that a manual override. So if you pick a fairly constrained region, algorithms like any algorithm that I've ever used, they sometimes struggle to find a good set of primers when you have a very narrow region. And so, like many of you, I'm sure I end up manually picking my primers anyway. And you can see that here's my primer sequence, and it's showing a primer feature, and these ends are extendable and retractable. And you'll notice that when I extend or contract, at the top here it tells me what my length is, what the TM is, and then I get this mispriming information, and we say bad if the energy is stronger than -4, and we'll say bad, meaning it's fairly strong. Doesn't always mean the primer isn't going to work. It just brings it to your attention that in this case we have a dimer that may be fairly strong with this set, and then there's pair dimers between the two primers, and then hairpins. But watch what I can do. I can move this around, this primer, and make manual adjustments on the primer. And so it's a really nice interface for just tweaking your primer a little bit and using your own experience, to pick what you think will be the best possible primer. And then I can work on the lower primer, click this button, brings me to the lower primer. Same thing, I can caterpillar this thing around and nudge it a little bit to give me what I want. So it's a really nice tool for designing that sort of a primer. And then of course I get a primer list, and this primer list then can be saved out, a primer catalog, and usually we just save this as a FASTA file or a tab delimited text file if I want to have columns of information. So that's how we can design primers to amplify across gaps. Once I do that, and I have a sequence, then I want to find out, how can I actually fill, a particular gap. And so I can bring the sequence into the project and try to direct it towards specific gaps. Now I'm going to show. So that's a de novo workflow. And in a genome where you don't have a reference sequence that you can use to assemble, you might not even be able to blast contigs to figure out, you might not know what the repetitive elements are at all, and you don't get a strong blast hit. And so you might have to do directed PCR to fill in these gaps. So a lot of work, but we have a nice interface for doing that sort of project. Okay, I'm going to move now from the de novo. We're going to do the gap closure in our other workflow. That's just what I have set up here. In our other work. Yes.
Before you move on to the next workflow, we do have a question about scaffolding. Is it possible to use PacBio reads to do the scaffolding if you don't have mate pair data?
We don't have an algorithm that will utilize the PacBio reads to, so you want to use the long PacBio read and then overlay contigs, to order the contigs using those longer reads, which I think is I think that's a strategy that is going to work. Our experience up to this date, though, is that the PacBio data, the error rate has been so high that we haven't been able to get enough high-quality data for the PacBio to actually do that sort of or take that sort of approach. That may change. We've spoken with PacBio people in the last couple of weeks, and we know that there's been some major improvements to the accuracy. If that's the case, then certainly we'd be able to take long reads, because they're long enough to stretch through the repetitive elements. And if they're more accurate, we'll just throw them in as part of the assembly. And then you don't really build as many scaffolds. You just simply have long reads that go right through the repetitive elements. And so that's what our hope is with the PacBio, this latest chemistry that I think it's in beta right now. But the older chemistry from earlier in 2012, it just is not accurate enough for us to make a good scaffold out of it.
Great, and kind of a related question. How do you bring sequence data into a previously run Denovo assembly? For example, if you have both Illumina MiSeq and HiSeq data for the same organism, is there a way to combine those?
So there isn't. So if you have raw sequence data sources, you'll want to use SeqMan Engine for that. And so you'll want to, in a Denovo assembly, you'll need to assemble those together. And so you have to start with SeqMan Engine. What you don't want to do is have a SeqMan project file that has contigs in it, and you don't want to dump in a huge Illumina file into that project. SeqMan Pro does have an alignment algorithm, but it's not suitable for the scales that you see with Illumina data.
Great, thank you.
So the other project type is the alignment to a reference genome with the automated closure, and that's what for the actual gap closure steps, we'll use one of these projects. So to get to that point, we have a genome assembly. We're going to pick the reference guided assembly with gap closure. And change the name of our project. And so I'm picking a K12 strain, MC106, as my reference. And I'm aligning MG1655 K12 data to it. So that's the project that we used in the last webinar to show this workflow. So that's, again, we'll use paired end data, same thing. You can see the interface is a little bit simpler here, fewer options, and it's ready to assemble. So when we have a reference sequence, we're going to use our high-capacity assembler. In about an hour, it'll go through about 3 rounds of assembly, the initial assembly to the reference, then the splitting of the reference at the structural variants, and then the de novo resolution of the novel areas. And so we actually get 3 project output types, one that is not processed at all, a middle project where there's intermediate processing, and then the final project, which is an editable SeqMan project that is in scaffolds, just like our de novo assembly. And so that's how we set the project up, and we'll take a look at one of these. And so I'll just diminish a couple of views here. So the project then, the scaffold just has this one contig in it. You can see it's 4.5 megabases long. So this is the entire genome. In the name, it says no split. So this is the first file. This is just the data aligned to the reference, no resolution or no splitting of the template sequence. And so if I look at a strategy view, I can see all the annotations on the reference. I see coverage, across the genome. And more importantly, I'm going to run a structural variation report. And so this is what you'll want to do if when you're starting out, you'll want to do the same workflow, look at this report, and see how many structural variants are present. Between these 2 K12 strains, there's 33 structural variants that we identify. Not all of them are true variants. Many of them are. Some of them are just some questionable areas in the assembly where it looks like something's happening. It might be an inversion or something that's not quite as clear-cut, but we split there anyway and allow the algorithm to resolve what that problem is. And so you'll want to look at this number and see, if it's over 300, then you're almost certainly going to want a de novo assembly or find a different reference. I think that the goal here, if you can find a reference where there's 100 or fewer, that almost always will be a better approach than a de novo assembly. And so this gives us an idea of what we have in store, how similar our strains are. And so that's one version of the assembly. And then we have, let's see here. And then we have the final output, which will have the word aligned in it, and it's a .sqd, which is the editable seqman format. And what we're looking at here, I'll just collapse this down, is we have scaffolds now, and these scaffolds are based off of the alignment to the reference genome, and we've automatically resolved many of the insertion sequences. So if I scroll up here, and again, it looks like the merging where we did contig align end-to-end in our de novo assembly. In this case, seqman engine used the pair information to fill in insertion areas and also just to automatically merge deletion areas. And so wherever it says merging in this report, that was the auto resolution of one of the gaps in your assembly. So what we could automatically do is merge most of the structural variants that were in our sequence strain. However, we're left over with a handful. So it didn't do all of them. We're left with 5 gaps here that we need to resolve. And so our output then is everything is in one scaffold. We have position information that's related to the reference genome, so we kind of know where this gap is occurring. And we also have unplaced contigs. These are contigs that, now I filtered out some of the small ones. So these are contigs that are long. They have good coverage. We look at them and, now they might have a thin end like that we trim off, but I scroll through and say, okay, that's a real contig. That goes somewhere in this assembly. I might do some editing here, though, and say, well, that tail, I don't like that. I'm going to trim it off. So we can do some editing here. So the contig pool is a place where we might, somewhere in these gaps, these contigs fill these gaps and close this genome. The reason that we don't have everything closed off is that if we look at these sites, like this, so this one is at roughly 66 kb. If we go back to our project, we can see that, at 62, we can see that at that position we have an insertion sequence. And knowing a little bit about this genome, this I believe is a prophage that's inserted into the genome. So it's fairly big, and that's the reason we didn't auto-resolve it. So what's generally left over here are larger insertion sequences. It can be phage that are inserted, ribosomal operon sometimes, things that were a little bit too large. We didn't have enough pair coverage to auto-resolve these. So we're left with a few gaps in the assembly. And so we need to figure out how to fill the gaps in. Now, one thing you can do is blast all the contigs here that are left over and blast the edges of the contigs. So I can go inside this contig. You want to select enough of it that you get through the repetitive, so it might be a couple hundred base pairs. And so I may blast the edge of a contig to try to get an idea of what genome element is at that edge. Because generally you sequence into it a short way, and you can figure out what's in that piece. And so I have a couple options here. I could export the consensus from the scaffold, like we did with the de novo assembly, and design primers and try to amplify across these gaps. So that's an approach I might take. Or I might do this blast and plug kind of an approach. And so, of course, I can go here and blast from seqman, and then look at my blast hits. And, of course, with E. coli, there's lots of blast hits. And my strain, that's my answer is here. So I know it's cheating. This will be harder for anyone with new strains. But I can see, here's a strain or a similar strain. Here's W3, a W strain here, where the range is almost identical. If you have strains that are fairly close in the database, you might be able to identify what that element is, and in fact go in and pluck out that piece from a reference genome and try to splice that piece in. So it's another way, in addition to PCR, if you can identify that this is, for instance, a prophage, I can grab that prophage from another genome that's closely related and plug it into the gap. And that's exactly what I'll do here, is if I think what's separating these two contigs is a prophage, I'll grab that prophage sequence and see if I can align it in. And so you'll notice there's another aspect to this project where I have these lock symbols. And it's another key aspect is when you have your contigs ordered into a scaffold, it's essential that you keep that order. You don't want to scramble up the contigs and lose the ordering. So when I'm adding new sequences, it's absolutely essential to lock the project down. I don't want sequences going non-specifically in the project. So if I've blasted my sequence and I've identified, okay, I think there's a prophage that is inserted here, and I've collected that sequence, I can go add it now. So I'm just going to do a sequence add, and I'll just go up here. So here's my genome closure project file and gap filling fragments. I may have PCR fragments, amplicons that I've generated and sequenced. I may have some de novo contigs that I'm working with. In this case, I have blast fragments, and I'm going to pick this prophage and add it into my project. So now I brought the prophage sequence into the project. And now what I need to do is I want to get this sequence into my scaffold. And so what I'm going to do is I'm going to assemble it in with everything locked down. So it's going to prevent it from using just a genome assembler from placing this in the wrong place. I just want to get the sequence into this project. And so there's a parameter that we need to change. If I go to project parameters, I need to change some of the assembling and pre-assembling defaults. The pre-assembling I have to change is don't add single sequence contigs. I want to add a single sequence contig in this case, so I'm going to change that default. And then the assembling, I'm going to switch over to the Pro assembler that has a higher capacity. It's just faster rather than the classic SeqMan. I might also change my minimum match percentage at some point, but in this case, I don't want it to overlap, so I'm just going to leave that. And then we're just going to assemble it in. And so because I have everything locked down, we'll just get our sequence here in our unlocated bin. Here's contig 64. Now, another thing that I might do is one of these contigs may actually closely match what I blasted and brought in. And in fact, so I could unlock, when I do that assembly, I could unlock some of the contigs and see if my blast sequence matches and it co-assembles. And in fact, the one that's almost the same size, contig 7, I know is part of the prophage. So I know it's really close. It's got a little bit of poisoned end on it that prevented it from going in. So I'm familiar enough with this gap that I know what we're dealing with here. But you can see they're almost the same size. I'm just going to lock this down, though. We're going to take our blast fragment. And here's how we move the sequence into, we just can click on it, drag and drop it, and now it's in the scaffold. The position is 0. So I need to, if I want to put it between the first and second contig, I can left click and hold and just type in a number that is between those numbers. So let's just do 25, for instance. And now when I sort, so now I've manually brought a sequence in, a blast sequence, into my scaffold in the right position. And now I need to use the algorithms in SeqMan to join these 3 contigs together into one contig and close this gap off. And here's where it can get, there's multiple options here. I'm going to show you the way that I brought the sequence in is the most foolproof option. And so now what I'm going to do is I'm going to unlock the contig. So I'm going to do one merger at a time, and I'm going to try to merge contig 64 with my reference contig. And so I'm going to select both of them, and then go to config, force join contigs. Now you'll notice there are some other enticing things that you might try. I might click reassemble config, or I might want to do align contigs. You definitely don't want to reassemble the contig. Again, this contig was assembled with SeqMan engine. There might be a million sequences in your contig if it's a big contig. You don't want to reassemble using SeqMan Pro. And I don't want to align contigs. Align contigs will flip contigs around. You could, reverse complement them, scramble your order. And we don't want to do align contigs end to end. That's something that we've done within the scaffold. So there's a number of options here, but force join really is the best interface and is what was designed for this kind of manual closure. Now, so the force join then shows us both our contigs, and here's the ends of both of them. I can grab them and slide. And so in some cases, you might know that these contigs overlap by 5 base pairs. And you might know that that's how they go together. So you could slide the sequences in place and force join them. So that's why it's called force join. It is a alignment independent way to merge these 2 contigs together. Or if you know the offset, if you know where they should overlap, you can type the offset in and then have them move to that offset and then align them. Or you can just use the aligning algorithm that's here, just a little pairwise aligner, and there it merged the 2 sequences. Because I didn't know what the offset was, or I didn't take time to figure out what it was. And so now I've merged, and you can see in the report, those 2 merged. There's a good overlap of 56 base pairs. And if I look at it in a strategy view, we can see what happened. And now the strategy view, again, this is a fully editable project, and so with lots of Illumina data, so it takes just a little bit to generate it.
Matt, while we're waiting for that, can you just clear up some of the terminology? We have a question. What is a scaffold?
So a scaffold is a collection of contigs. It can be anywhere from, it could be 1 contig, but usually it's multiple contigs, and they're placed in an order. And the order typically it can be based off of 3 things. Oftentimes it's based off of pair information. So it's the pairs that span across the gaps between 2 contigs. The forward and reverse pairs link up, and our software can put contigs in order, based on that pair information to build a scaffold. So a scaffold is going to have usually multiple contigs in a specific order that hopefully is the correct order in your genome. And so it's a way to organize contigs so that you can look at them in the proper order. If you didn't have a scaffold, for example, if I do a Denovo Assembly and I don't scaffold, I don't have pair information, and I get 250 contigs, without a way to create a scaffold, those contigs could be in any combination. And so the project becomes much, much more challenging to figure out what the actual order of those contigs are. And so that organization of contigs is really essential for gap closure. So here's the 2 merged contigs, and we can just see, here's the contig that has all the sequence data aligned, all the features from the reference sequence, and the thin red line is the piece that we just extended. And in the strategy view, I can see, here's my prophase sequence. I get this longer arrow. I can double click here. There's all my Illumina data. There's my prophase sequence. There's our overlap region. Okay, so that was the merger there. So half of the gap is closed in that case. So now if I want to join the next contig, I have to unlock it, and we can do the same kind of approach. Contig, force join, and again, I'm just going to align, and hopefully it goes together. And of course, this is where, if you blasted a sequence in, it might be from a close strain. You may have some mismatch, and this is where it does take trial and error to figure out, what the pieces are that close the gap off. so we'll let that. And this is a pairwise aligner, so it takes a few seconds to maybe up to a minute or so to actually go and run the alignment. And so now they've merged, and we can see we have one bigger contig now, and we'll get a strategy view. And so you keep doing this iteratively, for each gap. And sometimes the blast and fill works, but if it doesn't, if you get a region that's novel, then you may have to search in your contig list up above, and you might, bring a contig in or look at the contigs and see other areas that that aren't quite as accurate. Maybe if I edited these contigs a little bit manually, like I did earlier, if I look at a contig and see, oh, I've got one sequence extending off the end that might be poisoning the assembly, or a little bit of mismatch or ambiguous calls. So there's a number of different places we can go for those fragments, including this bin of contigs. Now you can see the strategy view. We've got here's our bigger gap, and that is our prophage, and then upstream somewhere here is our other sequence. I don't see it right offhand. So we can then look at the gap closure and see how we've merged our contigs together, and we can see the connecting contig, and again double click and go and look at and make sure there's not excessive mismatch. Now of course you need to verify when you plug the gaps. Now the next step here is to verify that in fact what I did is accurate and I didn't make a mistake. So what I'm going to do is just highlight this region, and then I'm just going to do a config save consensus as a single file, and I'm going to save this out as a gap. We'll just call it gap 1 sequence. And to verify that I did this correctly, I'll save this consensus out and then go back and realign my data. And if I splice these correctly, I should get even coverage, across these splice sites. So where I spliced in my gap filling sequence. If I see a lot of mismatch there or a big drop off in coverage, then I know I probably spliced something that didn't go together. I go back, I break it apart, and I try a different fragment. So that essentially becomes a step then, is you close a gap, I save out the sequence, I go back to SeqMan engine, so I go all the way back here, and it's a Genome assembly. Now I'm just going to do a templated assembly, and I would call this gap 1 test, and run a quick assembly that takes a few seconds, and now I add a gap filling sequence or consensus. Well, it looks like we're about out of time here. I know that was a lot of stuff to cover on something that's fairly technical. So I'll take questions here. I know that some have been emailed in. If we didn't answer your question in the webinar, I will certainly get back to you through email as well later today and tomorrow. So thank you.